Our next speaker is Dr. Vara Prasad. Dr. Prasad is currently a professor of crop ecophysiology and the director of Center for Sorghum Improvement in his Department of Agronomy at Kansas State. His research mainly focuses on understanding responses of food grain crops to changing environments and developing crop management strategies for efficient use of inputs. He is passionate about international research and education. He has several ongoing international research programs in Africa and Asia, focusing on soil, water, nutrient, and crop management practices, and stress tolerance for improving crop productivity. Dr. Prasad has published more than 100 peer-reviewed journal articles and book chapters and served as a peer reviewer for more than 35 different international journals. He currently serves on editorial boards of international journals, including Crop Science, Agronomy Journal, and Journal of Crop Improvement. All right, welcome, Dr. Prasad.
Can you all hear me? All right. Good morning. First of all, thank you very much for the Plant Science Council for inviting me to come here and share some of the things which I have done here in Florida and also continuing the same work in Kansas. And also thank you for the DuPont Pioneer for doing this, because this is a nice place where students can engage with scholars, and sometimes it changes their life, which has happened to me. Because when I was doing my bachelor's degree, it just took one visit to the International Crop Research Institute for Semi-Arid Tropics, ICRISAT, and that really got me excited about research, and I continued to do my master's there at that institute. And then where I met some international scientists in one of these type of settings, and then continued to do my PhD at the University of Reading. And then at Reading, I met Dr. Ken Booty at one of similar conferences, and I really thank him for giving me an opportunity to come here to Gainesville, University of Florida, as a postdoc, and then where I did a lot of research, which I'm going to show you here. And then at that time, he also connected with him and Dr. Hartwell Allen and Jim Jones, connected me with the international community they were working on in Ghana and Benin on peanut aflatoxin and peanut leaf spot disease. And then those are the contacts which made my life, made my career out of it. When I went to Kansas, I continued to do the same work, went deeper into the science, understanding the mechanisms, and then also wider, reaching out, going beyond West Africa. Now we work in several countries. So that's the impact of having this type of conferences where you have opportunity to meet with scholars, and persistence. In the morning, you were saying that persistence is one of the things which gets you there. So that's what makes the difference. So today, I'm going to talk about a particular topic, climate change and climate variability. Both the speakers in the morning really set the stage for me, because Dr. Sang really talked about climate change and food security, and then Dr. Green talked about the use of crop wild relatives. So this presentation is really a combination of both of those. I'm going to give you some specific examples of how climate change factors affect the productivity of the crops, and how we can explore the existing germplasm collections to really breed for tolerance. So I don't know if it was intentional or how the program came by, so it's really a nice segue to learn more about it. So I think I'm not going to spend a whole lot of time on some of the topics which both the speakers have covered, so I'll probably glance through them very quickly. I was told that you have to at least say 3 times to make you understand, or at least I need to be hearing the same message in different ways at least 3 times, so that I can make sense of it. So first, I think just 1 or 2 slides on the grand challenge and food production. And then what's the difference between climate change and climate variability? And then we'll talk about sensitive stages of crop growth and development, when the plants are exposed to shorter periods of stress, because sometimes you can just get 1 or 2 days of heat stress or cold stress, and is that enough to cause damage to the yields? And then we also look at longer term stress, how it affects crop productivity. And I'll show you a summary slide of mechanisms of reproductive failure. I'm not going to go into the details. I'll just give you a summary slide of that. And then there's a lot of questions which people ask, is it the daytime temperature or it's the nighttime temperature which is more critical? for crop growth and development? And how about the interactions between temperature and carbon dioxide? Dr. Sang showed you some examples of that. And then what's the interaction between temperature and drought stress? And there's a lot of unknowns. And then I'll end up with just showing you 3 or 4 examples of where we have used wild relatives, our genetic diversity, to breed for stress tolerance. So I would like to start with this hunger map, which Dr. Sang showed. Every day, about 800 million people across the globe go hungry or malnourished. And if you have heard about Norman Borlaug, he said that food is it's important, and agriculture comes first. So he said it's a primary requirement for our life. And all of his career, that's what he did. And when he started his career, when the Green Revolution happened, at that time, he was the only one person who saved 1 billion people through his research. So agricultural scientists play a significant role in food. So in some regions, especially in Africa, there's about 20% of the people who are undernourished. And Dr. Sang also mentioned about the overnourishment or obesity as well. And undernourishment is just one component of it. It leads to significant effects, especially in children. And then if you look at a lot of the work which is happening now, it's the first 1,000 days, which is very critical for kids. And sometimes that change is irreversible. So if you don't provide good nutritious food for the 1,000 days for the child, and that cognitive effects which happen during those periods, it's very difficult to recover from that effect. So it's very critical to take care of these children when they are young. So now the challenge is, as several people mentioned, that 9 billion people by 2050, and we have to feed them. And now we look at what the crop improvements have been happening in the recent years. We took care of that in the last, from 60s to 2000. The population increase and the food demand was met, primarily by bringing more land into agriculture, by improving the productivity, and then by more inputs. But unfortunately, we really don't have any of these options anymore. We don't have more land to bring into agriculture, or we don't want to do that because of the environmental consequences. And in some places, we really cannot intensify as well. We don't have the season to grow more than one crops. And then the efficient use of resources. That means you don't want to pollute the environment as well. So that's where the whole concept of sustainable intensification comes through. So this is an example of the genetic improvement which has happened in various crops. Wheat from 1965 to 1999, and then the green bar is from 2000 to 2008. If you look at this is a percentage increase. So it's been significantly decreased for wheat, rice, and soybeans, with an exception of maize, which has really remained at about 2.1%. And that's a direct implications for the investments of the private companies on that commodities. So the grand challenge is to produce more food from existing farmland. Dr. Prasad also mentioned about yield gaps. So there are opportunities. There's a tremendous amount of opportunities. If you look at this map, less than 30% of the attainable yield in Africa for most of the crops. So just to reiterate what he said, attainable yield and actual yield gap is the difference between the attainable yield and the actual yield, is what is called as yield gap. I'm calling it attainable yield. I'm not saying potential yield, because potential yield could be much higher. That's genetic potential. But remember, we can't, as Norman Borlaug said, we can't eat potential yield. It's the actual yield which is more important. So an attainable yield, the way I've defined it, is that it has been shown that some farmers, when they use the best management strategies, they are able to produce that. So it's been showcased by the farmers that you can attain that much yield in that environment. So when we do this yield gap analysis, it's critical to do the yield gap using attainable yields. And then again, the food waste or the yield losses. And on an average, about 30%. And then it varies. Sometimes they say that the more losses, more wastage or losses where. So in developing countries, it's more of food losses in the production and the processing, whereas in developed countries, it's more on the wastage side. So both developing and developed countries contribute to this food loss and food waste. So I won't go the details in there. I'll just go use some brief examples. Again, this was 2015 was the year when the carbon dioxide concentrations reached 400 ppm. And 2016 was the one entire year where the concentrations were more than 400 ppm. Okay? And that's also the case with other greenhouse gases. And then this is the temperature record. This is the temperature, climate change and climate variability. The warmest year in record is 2015. And that was about 0.7 degrees Celsius warmer. So certainly there has been a significant change in increasing temperatures all the way from 1980. It was pretty okay up until 1960, and thereafter it's been a significant increase. And there's also much more variability. So I want to make sure that you understand the difference between climate change and climate variability. Climate change is the mean things which happen over a longer period of time. And variability is that year-to-year variability or within the season variability. And if you are an agriculturist, it's the within the season variability which is probably much more important for your productivity. And that's where, because if you look, I'm in Kansas, so every time the summer comes, they say that it's the hottest summer. And then we come to the winter, and it's the coldest winter. So when you do the average, it really doesn't, there's no change. But the extreme events are the one which drives the productivity of the crop. I don't want to go into the details here. So, and in future, the greenhouse gas emissions are likely to continue to increase, and then that's going to lead to further more increases in the temperature, both in terms of the intensity of the temperature and also the extreme events. I'm going to skip this because the previous speakers already talked about it. But I would like to show one example here. This was a nice paper by these authors. They really analyzed the exposure of crop to critical high temperatures during the reproductive stages of crop development. I've done quite a bit of work on this, and then for most of the crops, the reproductive stages are much more sensitive than the vegetative stages. And then what they projected is that this one from 2000, 2030, 2050, this is the percentage of land under each of the crop, how much of that percentage of land will be exposed to these events. So they assume that by, right now there's about 19% of the land which has at least 1 day of heat stress. And then that is going to increase by 32% and 41% for wheat. And those are the difference for other crops as well. So it's likely that your crop is going to be exposed to shorter episodes of heat stress on a larger land area basis as well. So you may ask, 1 day, 2 days of heat stress at critical stages of crop development, would it make any difference? And you'll be surprised that yes, a day or 2 days of heat stress at sensitive stages can actually impact the yield significantly. So I've done quite a bit of work on identifying which stages of crops are more sensitive and what are the various thresholds. I'm just going to give you an example of wheat here. So most of this work was done in the controlled environments where we grow the plant under optimum temperatures, and then we move the plants and expose them to short episodes of heat stress at different stages of crop development and look at what impact does this have on yield and yield components. And the primary, I think most of you would know, what are the three components of yield? Yield components. Plant population, seed numbers, and seed weight. You multiply these three and you get your yield. So assuming that you have a nice plant population, the second biggest thing which drives is the seed numbers and the seed weight. And what the data which I'm going to show you is mostly on seed numbers and seed weight. And seed numbers are the result of effective fertilization, right? So for effective fertilization, you have to have the viable pollen and the viable stigma to have the fertilization process. And once the embryo is set, it is reasonably tolerant to stresses. It's very resilient. It will stay, and does not grow, but at least it is relatively more tolerant than the gametes itself. So this is the data where this 0 is anthesis, and this is 5 days, 10 days before, and 15 days before anthesis, and this is 5, 10, all the way up to 30 days after anthesis. This is a floret fertility. That's the percentage of flowers which were exposed to that heat stress convert into actually seeds. So that's that is highly correlated with the seed numbers. So as you can see, the lowest numbers, the highest dip is when the stress is happening at anthesis or just before anthesis. And that's true for most of the crops which I have worked on. I've worked on multiple crops, and the response is almost similar. So what does it tell us is there are two critical stages within that reproductive stage of converting from vegetative to reproduction all the way through physiological maturity. The two most significant stages which are highly sensitive is about 3 to 10 days before anthesis. And what are the things which are happening at that stage? It's the gametogenesis. It's the maturity of those gametes. That is, includes both pollen and the viable and the ovule. Both are developing. So any stress, even 1 or 2 days, this is 5 days. I'll show you data even for 1 day. So if you have stress during that period, it can significantly affect the viability of the pollen grains and also viability of the stigma. Okay? Here, that was 5 days on wheat. And then this is peanut, 1 day of heat stress, again anthesis, and about 4 to 5 days before anthesis. This is rice, 6 hours. Anthesis, about 9 to 12 days before anthesis. So these are the two major things which are affected. So I told you what's happening before anthesis. And then at the time of anthesis, what are the things which happen? Pollination, pollen tube growth, and fertilization. So those are the things which are affected at anthesis. And this is a direct example from this is in Australia, where this is a panicle, no seeds at all. This is from the farmer's field, and it came up in the news actually. And this is nicely set, and this is fully lost. And this is an example in Kansas, our own fields. Half of it is set, half of it is not. And you can directly correlate with the temperatures during that period of gametogenesis and pollination. I'm not going to go into the mechanisms, but I'll show you just briefly the pathway. Okay. So hopefully I've told you that the reproductive processes are affected. And I know there's a lot of students here who do genetics, molecular biology, and biochemistry. So I'm just going to give you a summary of that. So we know that high temperature stress in reproductive tissues causes decreased seed numbers. And that's primarily driven by decreased fertilization and pollen and ovule function. And I'm going to give you 5 mechanisms through which this process can actually happen. We have the data. If anybody is interested, just let me know, and I can give them those publications. The first one is the reactive oxygen species. So in general, plant has a nice balance of reactive oxygen species and the antioxidants under normal conditions. But when the stress happens, the reactive oxygen species goes up, and the antioxidant species comes down. There's an imbalance, and that causes the membranes to be damaged. That includes all the membranes like mitochondria, the thylakoids, and also pollen membranes. So those are affected. And the second one is the changes in the lipids. So because all the membranes are made up of phospholipids, so when you have this temperature increase, the composition of those membranes changes, and that damages the membranes as well and leads to the non-functionality of the gametes. And the third one is the pollen sugars. Right? So because for the functional sugars are the carbohydrates through which they get the energy, and the pollen sugars are decreased, and primarily because of the transporters. You may say that pollen is a very small cell, and plant has tremendous amount of leaf, and there's so much of carbohydrates produced, and how come a small pollen is viable, non-viable because of lack of sugars? The reason is that anther wall is full of sugars, but it's the transporters which move that carbohydrates from the anther wall into the pollen grain. Those are affected by the stress. So when you don't have those transporters, although the plant and the anther may have a lot of carbohydrates, but it's just not going into the pollen grains. So that's the reason for loss of that. And then anatomical changes are also happening with the heat stress. The anatomy of both pollen grains, especially the thickness of the externe wall, which determines how long the pollen is going to be viable, is affected by heat stress. And same is true for stigma and pistil as well. So you see those. So those are the primary 4 reasons. And the last one is the dehiscence. Because although it may have a nice viable pollen, but if the conditions are not good for dehiscence, and if the pollen actually does not fall on the stigma, there is a difference of the height difference between the pollen and the stigma, then the fertilization is not going to happen. So these are the primary mechanisms through which high temperature affects and leads to lack of fertility. And then the next question people ask is that whether it's the pollen or the stigma which is more sensitive. And that's a very common question, and it's when students asked, and I really had no answer for that. So we were looking for a system where we can actually test that. And sorghum, because of the cytoplasmic male sterility, is a nice system to actually test that process. So we did a lot of experiments where we had both male and the female exposed to normal conditions, and then the male exposed to high temperatures and female normal. So we tried to cross multiple combinations. And this was, when we tried, this was too extreme. This was 38. So even at that temperatures, in general, pollen was more sensitive than the stigma, and the combination of those was the worst. Then I thought, well, does that happen in all the crop species? It's probably not, because this is on sorghum. In pearl millet, it's actually the opposite. In pearl millet, we had stigma. We had the female, which is more sensitive than the male. Combination is all bad, and the reason being, when we looked at what might be the reason, because pearl millet is a protogynous, so it produces the stigma first, and then the stigma exudes out and is directly exposed to the temperatures. That's only crop which, at least from the grains perspective, which have that type of thing. Whereas wheat, sorghum, rice, it's different. They're all the stigma does not protrude out. They're not protogynous. Sorry, I think I should have done that first, but I'll get there. So that was about the effect of short season temperature stress, or short episodes of heat stress. Now, season-long effects, right? So each crop species has an optimum temperature and a ceiling temperature at which it really does not produce anymore. And there is a huge difference within cereals and also within legumes. And this work is a summary of what I've done at irrigation part here with Dr. Booty and Dr. Hartwell Allen and their past work. So we now have a summary from all the major cereals and all the major legumes. So this is, I chose to show you harvest index because that captures the effects during the entire season in all processes. So as you can see, this is a mean daily temperature. That means the maximum temperature plus the average of maximum temperature and minimum temperature, which is daytime high and nighttime minimum. So if you look at this is a wheat. So the ceiling is 31. This is rice, the blue one, 35. Sorghum is 38. Pearl millet is 41. So there is differences among within the cereals itself. So it would change. The farmers will have options, but they'll have to pick the crop which is more suitable for their area. And within legumes, the dry bean is 32, peanut is 39, and soybean is 40. So there is that is crop species variability. And if you look at where these crops are grown now and what are the conditions there, most of these crop species are already growing in a region where it's just about optimum. So any further increase in the mean temperature, we're going to be looking at decreasing those yields. But there are maybe places where we have to shift the crops. Some places it might be better to grow wheat, and some places it might be better to grow sorghum and millet. And we need to be able to make those changes to move from one crop to another. So there are opportunities there. And I'm not going to go into the details here. The second question which people ask me is it the daytime temperature or the nighttime temperature which is more critical? And in general, the climate simulation models project that the nighttime temperatures are going to be going up faster than the daytime temperatures. So now, which one has more effect on which of these processes? So I've done quite a bit of work on that as well. This is the wheat. This is just response to nighttime temperatures. Just effect of nighttime. Just look at this reproductive fertility. With increasing nighttime temperature, there is decrease. And then individual grain weight also decreases. Remember the components of yield, right? Seed number, seed weight, both of them are decreasing with increasing. This is only high nighttime temperature. We kept the daytime temperature very constant in this one. And what is the one which is driving the, we saw this decreased grain weight, right? Individual grain weight. What drives it? It is because individual grain weight is a product of the duration, the time the seed has to fill, and the rate at which it is filling. So if you multiply those two, you get the seed weight. So now, this is the time after flowering, and this is a rate. The slope of that curve is the rate, and the place where it intersects with the x-axis is the duration. So with increasing nighttime temperature, the duration is decreased. But whereas there is not enough change in the rate. There is slight increase in the rate here, but it's not enough to compensate for the loss which is caused by shortened duration. So the primary reason for decreased seed weight, individual seed weight, is decreasing in the grain filling duration. Okay? That's what is driving it. So now, this is an example where it shows different temperatures, optimum, high nighttime temperature, high daytime, and the combination of those. So again, this is mostly from brooding to maturity, so it really captures the entire reproductive phase. And there's really no clear consensus. Pretty much high nighttime and high daytime are similar effects on spikelet fertility, whereas in the grain numbers, it's a high nighttime temperature was not as bad as we thought for this particular wheat, but in rice it's the opposite. There's a clear more negative effects of high nighttime temperature when compared to the daytime temperature. So it's really not clear what's the thing which is driving. And what comes to my mind is the respiration, because respiration is a deal which happens 24 hours of the day, and then photosynthesis is only half of the time, and then the gametogenesis, which is the development of the pollen, which is 24 hours, whereas the pollination and fertilization is only 4-6 hours, because pollination happens early morning and then fertilization is finished by noon, whereas gametogenesis is happening 24 hours. So based on the timing and the sensitivity of those, that's what drives whether it's the daytime or the nighttime temperature, which is critical. And Dr. Assange also talked about when we say climate change, increasing CO2, we say increasing CO2 is very good because Rubisco is more affected, and then there will be more photosynthesis. That's true for C3 crops, and to some extent, some C4 crops also, we see about 5 to 10% benefits of that. But greenhouse gas, CO2 has its own capacity to trap heat, so that will cause the temperature increase. And then, because of this stomatal closure and the elevated CO2, the tissue temperatures will go up. So what we have seen here, this is work done at University of Florida. At cooler temperature, you see benefits of elevated CO2. But when the temperature goes up, you completely lose that benefit, and sometimes it's even more negative. And that's true for a C3 species like rice, and that's also true for C4 species for like sorghum. So all the beneficial effects of CO2 only happen when the temperatures are optimum or cooler. The high temperature overrides the benefits of CO2. So if you are in the grain production business, then that's bad. But if you are in the vegetable production, leafy vegetables, or in forages, you're okay. Because photosynthesis is not affected by heat stress as much as the reproductive processes are. The final one is the interaction between temperature and drought. Because, you know, those are the two major things which would happen, increasing in temperature and then increasing in drought. So is there an interaction? Are the processes same? Are the processes different? And is there an interaction between them? And most of the time, in places where you do not have irrigation facilities to overcome, these go hand in hand. So dry and heat will always be hand in hand unless you have the irrigation facilities. So here's an example. Just focus on any one of these left side graphs, okay? So this is optimum, both irrigated and optimum temperature. And this one is high temperature. And this is drought, and this is a combination. So in this case, this is on wheat. So we saw that heat has more effect than drought. But again, it's difficult to quantify drought because it's different soils and then intermediate rainfall and how deep your roots go. So there's a lot of complexities there. But the thing is that it's an additive effect. So if you have drought and heat, it's not that the combination of those would be lower than any of these two events. It's generally additive or even worse. So now the last step, last one. I don't want to just show the effects and say what can we do as breeders. And Dr. Green really showed a nice examples of reaching out to wild germplasm or the germplasm resources. And if I can just talk about sorghum. Sorghum has about 45,000 accessions. And only about 3 or 4% of that is actually utilized, either in the plant breeding or for any of the stress tolerance. And more so, the germplasm resources have been utilized for biotic stresses, but not so much on abiotic stresses. So I'll just show you some examples from wheat. Well, actually, wheat and rice. So before I go there, I'm a crop physiologist, and most of the time when physiologists say, we talk about so many traits, and breeders gets confused, and breeders say, well, I'm not picking any of those. So we really have to do a targeted effort to really showcase that these are the traits which are valuable, so that breeder can actually go ahead and evaluate them. And same thing is also true for giving something to agronomists as well. So, and then our farmers at the end. So I would like to go back to the yield components. If you're working on a trait or any mechanism which does not affect directly the 3 yield components, which is the plant population or the biomass, seed numbers, and seed weight, you rethink about it. Because the process which will take you to move to the farmer's field is going to be much longer. But if your trait directly affects any of these 3 components, then you have a better chance of success, especially as a PhD students or master students who really want to deliver something and have an impact and publish, or really see where your work is directly relevant to the farmers. So I would start there. Just do an initial evaluation. Does it affect biomass? Does it affect the plant population? Does it affect the seed numbers? Does it affect the seed size? If not, just think how can you actually make that happen. So I'm going to show you 4 examples. Reproductive success, because I told that's very important, and that's the one which drives the seed number. And then another trait which I'll say is time of day of flowering, which also affects seed numbers. What do I mean by time of day of flowering? In a day, generally the temperature starts to go up from the morning, reaches peak about solar noon or noon to 2 or 3 p.m. Based on where you are, and then it drops down. And I told you that the gametogenesis or the pollination is more sensitive, but once the embryo is formed, it's relatively more tolerant. So if you have a variety or a plant which can fertilize in the night when the temperatures are cooler, or fertilize very early in the morning and finish up that formation of the zygote and then stay calm, then it has a better chance of success for seed numbers, right? So you don't want to be anthissing and dehiscing and pollinating at the hottest period of the day. And then the favorable respiration, because growth is the difference between the photosynthesis and the respiration. The remaining carbon is what is used for the growth. So most of the time we always talk about increasing the photosynthesis, but we have not really found first of all, it's difficult to measure respiration. And then even if you try and do it, is there an optimum respiration or not? That trait has not been really utilized. And even if you look at photosynthesis, I don't think there is enough genetic variation for photosynthetic capacity itself for any of the crop species. So that needs to be explored as well. We'll mostly talk about environmental effects on photosynthesis, but not so much in genetic variation as well. And the last one is, as I said, seed size is a product of grain filling duration and grain filling rate. So is there any genetic variability? Or even if it is, does it compensate with each other or not? So looking for genetic variability for seed filling rate and at the same time having a longer seed filling duration would be valuable. Generally what happens is if your duration is smaller, you fill it up at a faster rate and your seed size is the same. Or if you have a longer duration, you fill it up at a slower rate and your end product is the same. So you really look for something which has higher seed filling rate and also longer seed filling duration. So those are the traits which we will be looking for. Here's an example for seed set, some tolerant lines, N22, which was really setting more seeds, and compared to those which were not. So that's an example of seed set in rice. And then early morning flowering is again a wild glaberimus. Rhiza glaberimus was flowering early morning, whereas the sativas are all flowering at late in the morning and also in the noon time. So that's just one example where we can go to the wild species. Pearl millet, most of the anthesis happens in the night, and fertilization is done in the night. Cowpea, also some of those. So those are the crops which are much more resilient than soybeans or maize or rice. And another area which we have explored is wheat wild relatives. We looked at multiple species, Speltoides geniculata, Cirsa and Longishima, and there was some genetic variability there as well. And we were trying to link if that genetic variability is associated with the place of origin or where they were adopted. And sometimes there is clear evidence, sometimes there isn't. So it was very difficult to say that, okay, if you go to a hottest place where the crop originated and capture from that, you might have a more tolerance. But it was not always the case. So it's not always true. I'm going to skip this, okay? So hopefully I have convinced you that climate change and climate variability will have negative impacts on grain yield, especially on the food grain crops. And yes, short episodes, as small as 6 hours to 3 days, can actually negatively influence yield, and primarily driven by seed numbers, which is driven by the viability of the pollen grains and the stigma. And then the most important stage within the reproductive stages is the gametogenesis, flowering, and the early seed filling stages. And then the beneficial effects of CO2 are negated, more than negated, by high temperatures. And then in the existing germplasm, which the breeders already are using, there is not much genetic variability. So we really need to go out for the larger germplasm collections on the wild relatives to capture those. And then the one piece which is really missing, or I haven't had a chance to talk about, is the phenotyping. Because genotyping has made significant progress in the last 10, 15 years. But there is not, especially because of the technologies which were driven primarily by the private companies. But phenotyping is really the constraint at this point in time. To phenotype 1,000 to 2,000 lines, if you go out to measure any of these traits physically by students or undergrads or graduate students, is going to be very difficult. So that's the biggest gap at this point, is developing high throughput phenotyping. And sometimes it's difficult to get high throughput phenotyping directly for that trait. So we need to find surrogates or strong correlation between some of the traits which could be captured using UAV, unmanned aerial vehicles, from a larger picture spectral imaging or thermal imaging, and then try to correlate with one of your traits and then see if that works. I mean, there's a lot of work which is going on now. I think that's where the next step is going to be, high throughput phenotyping, and a lot of companies are also investing on that, and we're going to get it. So that's the one which is there, opportunities there. And then the last one is we only talk about individual stresses, and we have done about temperature, we've done about drought, and maybe drought and CO2, temperature and CO2, but abiotic and biotic stresses interaction, that's a huge gap. There is not enough literature looking at the interactions of abiotic and biotic stresses. So you can make a career out of it, as I made my career from heat stress alone. So I think there is a lot of things to say here. And then I would like to end with just saying one thing is that it changed my philosophy when I first came to Dr. Booty's lab in 2000, exactly in 2000, yeah. So because when we are a graduate students and when we are looking for master's PhD, we dig deeper, and we specialize in then one particular area. But to reach farmers, it's a system. Farmer is not just going to pick one of your trait or one of your things. It's a system. So, and it takes multidisciplinary approaches to address a solution. Because let's say if I said pollen viability, right, just as an example. Now, I go to the breeder and say, well, how do I measure pollen viability on 2,000 lines? It's impossible. He's going to give up. So we really need to find out whether is that pollen viability related to seed numbers? Is the seed numbers related to something else? So it really takes multiple disciplines to address a solution. And the same thing is true for droughts. I'll give you an example of drought stress. A limited transpiration work which Tom Sinclair did, also from University of Florida. So having to go and measure these limited transpiration traits, get that curve on, you can't get more than 10, 15, 20 lines, you can do it, but if you're talking about 2,000 lines, how do we get that? So the alternative is that think about limited transpiration. What would it do? It could change the canopy temperature, tissue temperatures, right? So can we measure the tissue temperature using a UAV? And because even if you're doing handheld, from 5 minutes to 5 minutes, the temperature changes, the wind changes, the humidity changes. So you really have to work with the engineering people to see whether you can capture that thermal image from UAVs. So what I'm trying to get to is that multidisciplinary work is very important. And as a physiologist, or even as a molecular biologist, to reach the farmers, we have to cut through breeders. Because I can't have a direct impact on farmers' field. I have to work through breeders or agronomists. So I better make sure that what I'm working is valuable to the breeders and valuable to the agronomists if I want to see my work to be directly effective in the farmer's field. And as Dr. Burlock said, take it to the farm. There's no point in just doing all the work in the laboratories. You can do excellent work in laboratories, but it has to, at some point of time, should be taken to the farm, and you better do it at early stage, because what works in laboratories may not work in the field. So you just want to give it a try, see if it works, and then continue to dig deeper so that you can have a better impact. With that, I'll thank you very much for giving this opportunity. Yes.
Yeah, I was thinking about your comments about the time of fertilization, how long that takes. At least in corn, I mean, the pollen has to shed, it has to germinate on the cells, it's about a day for the pollen to grow, and then affect fertilization. So we don't think of it in terms of a few hours per day. We think of it as a 24-hour cycle. Is it fundamentally different in the small grains? I mean, how much time between pollen shed to the double fertilization?
That's right. There's a huge species variability. If it is rice, it's about 30 minutes to an hour, 30 minutes to an hour. Yeah. And peanuts at about 6 hours. Cotton, it's 24 hours. And then I'm assuming it's for, I haven't worked on maize, but it's probably more than 24 hours. Yeah. But so that varies from species to species. And more important thing is that once it sheds, whether the stigma is ready or it provides the support to the pollen grains. If that's not happening, then even if it is viable, it's just going to lose it. So in case of silks, I think it's probably the contact and enough energy or enough signals are being produced by the silk so that it continues to grow. The pollen tube continues to grow. Yes, sir.
Anyone screen for pollen specific? You seem to allude to it as though there's not much variability.
Well, I think I don't have a direct answer for that, but at least in terms of our experience, there is really not much differences in photosynthetic capacity across various genotypes within a species. Even under optimal conditions, you don't have that variability. But once you do under stress conditions, it may be the effect of that stress which is giving that differences, but not the innate photosynthetic capacity itself. At least for the crops which we have, we've done it quite a bit on peanuts and quite a bit on sorghum and wheat. I haven't really seen a significant difference in photosynthetic capacity or photosynthetic rates at optimal conditions. Under stress, yes, but that's the effect of stress itself. Yes, Charlie. Yeah, Charlie. Yeah. After that, maybe.
This is about, you mentioned the sugar transport.
Yes, yeah.
I was curious which ones, in corn you said, well,
yeah.
pollen and
Yeah.
the same, or how you compare with others?
That's right. So the work which we have some work from sorghum done also with Dr. Budis and Jan. It's a hexose transporters. So it's one hexose transporters are the one which are downregulated under heat stress, and those are the ones which are affected. So it's a particularly hexose transporters. Yes.
Your arguments about which is more important, day temperature, night temperature. I mean, the answer you get is totally dependent on the range you select.
Yeah, absolutely, yeah.
And so, what was the rationale for and I forget now what the range is for, but
yeah,
the day range and the night range?
Yeah, in general, you have a much more wider ranges in the day for temperature variability, not so much in the night. But even a slight increase in the night temperature would have major effect on the yield components. The primary reasons being, I gave an example of both, let's say, respiration, photosynthesis, and gametogenesis.
But what was the rationale for the range that you picked? I mean, I agree with what
Yeah.
But did you look at optimal versus predicted versus...
Yeah, no, I think we went very extreme ranges. I think for that we went almost from 18 degrees Celsius to 24 or 28 degrees Celsius. But those nighttime temperatures do exist. Like, for example, if you are in the tropics area, your diurnal variability is less. Whereas if you go to much more like Kansas or somewhere else, you have the high daytime temperatures, but also you have cooler nights. So when you look at, and I haven't shown the example, but there is enough evidence that you may have the same mean temperature. But if you have this amplitude difference between day and night, if you have high day and cooler nights, the effect is not as dramatic as you have high night but cooler days. So the amplitude, I have published the paper in Crop Science, I think, so on maize itself. So 18 degrees diurnal difference versus multiple difference. So as the diurnal difference becomes less, the effects are more severe, and that's primarily driven by the nighttime temperature.
Thank you, Dr. Prasad.
Thank you.